All right, our next speaker is Dr. Muhammed Murtaza. He will talk about circulating tumor DNA, and this is really an important topic. It's all over the news. He is one of our colleagues here at the Mayo Clinic in Arizona, also has an appointment at TGen. He actually did all his fancy studying, his PhD work at Cambridge, and is actually was really the primary author of the first circulating tumor DNA paper that came out in the New England Journal of Medicine. So we're extraordinarily lucky to have him.
Thank you very much for that introduction, Barb. Thank you to the organizers for having me here and speak. And then thanks for the opportunity to share the work. These are my disclosures. I am interested in developing circulating tumor DNA analysis, so I have relevant IP, which is licensed to some of these companies that are taking them forward. Okay, so my objectives for the day are really threefold. Start with identifying sort of where the potential utility and applications of circulating tumor DNA analysis are in metastatic and localized breast cancers. I think the number 2 point is perhaps the most important, clarify aspects of ctDNA analysis that are ready for or at least clinical studies or closer to clinical utility, and then also recognize limitations. So if I was to reword this, we look at a number of things, and then hopefully, we can figure out what is still hype and needs to be tested, and what may be sort of more relevant and we should start to use it, or at least think about it. And then describe some of the ongoing research that we've been doing in ctDNA analysis for treatment monitoring in early-stage breast cancer. So for almost 6 decades now, we've known that if you take a blood sample and you spin it down and you get rid of all the cells, you're left with cell-free plasma, and there are DNA fragments that are floating around in plasma. For about 2 decades, we've started to realize that if you take this blood sample from a pregnant mother, then a fraction of this DNA is actually specific to the fetus, which has led to the development and now clinical implementation of non-invasive prenatal diagnostics and screening. The analogy here is in cancer, where in a cancer patient, a fraction of these fragments are cancer-specific, are shed by the tumor itself, and you can tell them to be cancer-specific because you can find the same somatic mutations that you would find in the cancer in these DNA fragments. So whether they're point mutations or amplifications and deletions, the same things that you would generally find in a tumor biopsy using a sequencing assay, you can actually start to find in plasma samples. So the field of